Hello, everyone, and thank you for joining us for today's webinar, Enabling Complexity Drug Substance Manufacturing via Continuous Flow. I'm Heather Bisaccio, your host from Life Science Connect webinar series. With us today, we have Dr. Matthew Bio, Chief Scientific Officer at Cambrex. Over the next hour, he'll be discussing examples of the use of flow technology to enable modern synthetic methods from pilot scale through commercial. After his presentation, we'll be holding a live Q&A. Feel free to submit questions as we go along. We'll get to as many as possible during the event, and any that we can't get to, we will follow up with after the event. If you run into any issues, please try refreshing your browser first. If you still need additional support, go ahead and submit a question via the Q&A, and I'll get you back on track. Finally, the webinar will be available for on-demand viewing. You'll receive a link via email as soon as it's available. With all that said, I'm going to turn it over to Dr. Bio to get us started.
Hi, thank you very much, and thanks for attending today. It's my pleasure to tell you a little bit about some of the advanced technologies at Cambrex, and specifically today about continuous manufacturing. So Cambrex is a network of 13 sites across Western Europe and the US. Our primary manufacturing sites are in Charles City, Iowa. High Point, North Carolina is a commercial site for small volume commercial. Milan, Italy, and Karlskoga, Sweden. We have a number of specialty sites as well in Wiesbaden, Germany, focused on biocatalysis, Edinburgh, UK, which focuses on solid state characterization, the Snapdragon Chemistry site at Waltham, Massachusetts, which is a center of process development for continuous manufacturing and complex synthetics. Durham, North Carolina is a site of analytical excellence with extensive capabilities in drug substance and drug product characterization, including biologics. And then Longmont, Colorado is a site that's dedicated to early phase development, including drug substance and drug product manufacturing at the same site. Today, I'd like to tell you a little bit about the advanced technologies that we are able to deploy, specifically continuous manufacturing. But first, I'd like to highlight that at Cambrex, we're able to handle the entire life cycle of CMC for pharmaceutics, from pre-IND and IND enabling services, including solid state characterization, standalone analytical services, first GMP deliveries, GLP tox deliveries, formulation development, and all your stability and storage needs. And then through life cycle into commercial, we're able to manage GMP manufacturing at very large scale. Our largest vessels at Charles City go up to 16,000 liters, where we have 13 work centers that each have 16,000 liter capacity as their top end reactor. Along with that, we're able to handle the full suite of process characterization, development, and validation activities, including analytical validation. And we do this all in a sustainable manner. Today, I'll focus on our capabilities around continuous flow, but I'd also highlight we have the ability to handle potent molecules down to OEB5 and controlled substances across the network. At a later webinar, we'll discuss complex synthetics, things such as peptides and oligonucleotides. And separately, we'll have a webinar in the future on biocatalysis capabilities at Cambrex. But today, we're going to focus on continuous flow, R&D, and manufacturing. Cambrex has extensive experience in continuous manufacturing. We've chosen to break those out into platform technologies that we can reliably translate from laboratory scale through to production. We've separated them into the classic reactor types, plug flow, open tube or static mixed tubular reactor, typically used for fast cryogenic chemistries, liquid gas chemistries, and hazardous reagent management, such as azotizations, diazotizations. Packed bed. This is a system where there would be a solid phase catalyst or reagent over which either a liquid-liquid or liquid gas reagents would flow. A continuously stirred tank or CSTR. These are typically deployed when there's heterogeneous type chemistry where you'd have solids present, and have found particular value in the generation of Grignards from base metal magnesium, the generation of organolithiums from base lithium metal, and the generation of organozincs from zinc metal. In addition, they're also has utility in enzyme catalyzed reactions and cross couplings involving solid bases. We have 3 specialty reactors that we've developed, or capabilities that we've developed as well, around photochemistry, with the full range of photochemistry from Photoredox all the way through in the 400 nanometer range down to even short wavelength, 254 nanometer wavelength capabilities, and all the way out to 660 nanometers, where you might do singlet oxygen type chemistry. In the space of electrochemistry, we've published in this area and also have examples ongoing in terms of cross-coupling and the use of this technology for oxidation and reduction type processes. And then finally, in terms of reactions, we have published a few years ago on a novel use of evaporation membranes to affect ring closing metathesis, where ethylene is permeated through a membrane continuously during the course of the reaction. In addition to reaction unit operations, we also have unit operations downstream for workup and isolation available as continuous platforms, including aqueous workup, liquid gas separations, continuous distillation, crystallization, and filtration. Today, I'll focus on 2 areas in particular. One will be an organometallics project that was performed in continuous manufacturing, and the other is a photochemistry project that was performed in flow. So for the first case study, we'll discuss an organometallic project that involved alpha lithiated carbamates. So back in the 80s, Peter Beek and co-workers developed a novel lithiation of these Boc-protected amines. This one shown here is the dimethylamine, but it's been applied more widely to other substituted amines as well, such as the pyrrolidine most commonly. In this reaction, the Boc group stabilizes the lithiation of the methyl group on the nitrogen, or this could be a CH2 group, to generate this stabilized organolithium, somewhat stabilized, that can then be utilized with a number of electrophiles and engage in cross couplings as well to affect synthesis of some complex amine Boc-protected amides, as you see here in these bond disconnects, where the red bond is indicative of the bond that's formed. So from these simple Boc amides with sec-butyllithium and TMEDA, you could then access and build up a broad range of amines for use in downstream synthesis. So in the case of this particular reaction, we were interested in obtaining this lithiated Boc amide so that we could then add it to a carbonyl electrophile. The way that this was developed in the laboratory is there were 2 cryogenic reactors side by side. In the first one would be the Boc amide, TMEDA, and to that would be added sec-butyllithium at less than minus 70 degrees. After some period of time, that lithiated amide that's formed would then be transferred, again under cryo conditions, to another cryoreactor, also held at less than minus 70 degrees, holding the lactone. That would then be product forming and go on to quench. This worked well in the laboratory scale, achieving yields of up to 70%. However, on scaling, even to 1 to 2 kilos, you would get reduced yield down to 50 or 60% at 1 to 2 kilos. And then scaling beyond that was deemed impractical because of the decreasing yields that were observed as we went up in scale. Beyond the yield implications, the complexity of having 2 cryoreactors in tandem and then being able to transfer from one to the other while maintaining low temperatures was also quite problematic. In studying this reaction gave insight into why the yield was decreasing over time. As we went up in scale, cycle times would get longer and the low temperatures required were more difficult to achieve in batch reactors. So using ReactIR, we can see all the species of this reaction. So the large peak being the Boc amide, which then disappears in a dose-controlled manner by during addition of sec-butyllithium, resulting in the lithium amide, which can also be visualized by IR, which is then quenched by addition of the carbonyl compound and consumed to make product 4. What we noticed in this is that in addition, the lithium amide actually shows competitive decomposition even at -78. So in this cartoon, what you see is sort of the situation that's going on in the flask. As we're adding sec-butyllithium to the reagent or to the Boc-amide, it's getting lithiated. At the same time, some of that lithiated species is decomposing. So if we run the reaction to incomplete conversion so that we can use the maximum yield of the Boc-amide, lithiated Boc-amide, then we run the risk of transferring sec-butyllithium into the next step of the reaction, which would react with the carbonyl. If alternatively we wait until all of the sec-butyllithium is consumed, then we're also in a regime where the lithiated Boc-amide is decomposing. So what's needed is precise control over the timing of this reaction so that we can be within this green box of ideal residence time where the yield of 2 is maximized while the concentration of sec-butyllithium is reduced to a minimum. And this is an excellent case for flow. This ability to precisely control the timing of the reaction is exactly where one would want to apply continuous manufacturing. So the characteristics of this reaction are that it's a fast reaction. And the reactor characterization in plug flow is that would have rapid mixing to enable that fast reaction to be in the right stoichiometry. And I'll talk about that in a minute. It's a highly exothermic reaction. So high heat transfer efficiency is very important. As we go up in scale in batch, it gets harder and harder to get heat out of that system because the surface area obviously is decreasing relative to the volume every time we go up in scale. And that makes it more and more difficult to get that heat out of the system. So maintaining -70 degrees gets more and more challenging as we go up in scale. We have unstable intermediates that require precise control over reaction time, which is a reactor characteristic for plug flow. And it's also stoichiometry sensitive. And so I'll talk a little bit about the precision that's available in terms of the mixing control over a plug flow reactor and how that plays in here. So about, I would say, maybe 15 years ago, we started to get involved in continuous manufacturing pretty heavily. And many years ago, when we were discussing mixing in these systems, I really was having a difficult time understanding the implications of laminar flow. So laminar flow is the situation that we find in most plug flow reactors where there's no turbulence in the mixing. And so when you bring two streams together, they actually run parallel next to each other. And what you see is just mixing by diffusion. And so I needed to be taught about this and I needed to visualize it. And if you look on YouTube, there's a number of really excellent visualizations of this, but this is the one that really struck me. So what we have here is we're taking a solution of phenanthroline with 0.1 molar hydroxide. And that's the pink solution coming in from the bottom of this tea mixer. And then we're adding to it a slight excess of HCl, 0.11 molar HCl. And what you would expect if these were completely mixed was that what comes out of that tea would be a colorless solution. But instead, what we see is a pink solution persist along the length of that tube there, about 20 centimeters. And if you actually were able to get in very close, you could see that there's actually two layers, one clear layer and one pink layer. And that clear layer being the 1 molar or 0.11 molar HCl that's being added. So this is quite striking because you'd think that this mixer is accomplishing the mixing and you've got these two fluids that are both in the same solution solvent, and why aren't they completely mixed? And this is the consequence of laminar flow. So the situation gets even worse when you're doing organometallic chemistry because now you have a situation where these two streams are moving next to each other. And this is my crude cartoon up in the corner where you've got your organometallic in red and you've got your aryl halide in blue. And what happens is you're relying only on diffusion for these to mix. So a little bit of it will diffuse, of the aryl X will diffuse into the organometallic phase and vice versa. And if your aryl X electrophile has more than one reactive species on it, it's now found itself in a sea of organometallic. And since that reaction is very fast, it's not going to wait around for diffusion until it reacts and it's completely mixed. It's going to react immediately and see, because it sees a huge stoichiometry of this organometallic. So what's the solution to this? So a very simple and old technology is static mixers. And what you see here in this enlargement is the helical static mixer put down this tube. And now when you do the same experiment with this, you see that the pink color is gone almost immediately after it exits that mixer and is crossing that static mixer. And you can actually use this information to characterize mixing across these static mixers. And you can do it across scales as well. So what we have here is with that well-known reaction of acid-base, the kinetics are well understood, and there's a number of other reactions that can be used as well that have well understood kinetics to figure out reaction timing. So in a very simple way, we can just use that color disappearance to determine the mixing completion time for the reaction as it travels across the static mixer. And in this case, I've got two static mixers on the side here shown, one at 1.7 millimeters and one at 3.75 millimeters. And we've also characterized them out to 6 millimeters. And you might look at that and say there's not much difference between those in terms of size, but in terms of productivity, when we're talking about flow rates on the order of 10 mils per minute or above, that 1.7 millimeter reactor is actually producing hundreds of grams per hour. That 3.75 millimeter reaction with this fast type of chemistry can produce kilos per hour. And once you're out at 6 millimeter, you're talking about tens of kilos per hour or beyond. So these static mixers are very low cost, operationally simple mixers that are essentially can be disposable or easily cleaned. They can be well characterized by this example where you can use a known reaction to characterize mixing completion time. And that means that in laboratory scale, we can assign a mixing completion time that hits our target product quality profile. And we can translate that by model to larger scales because we've characterized mixing in those systems. And if the mixing of a helical mixer is not enough, there's more advanced type of mixers like these X-grid mixers that have even faster mixing times available. I would contrast this with dynamic mixers, which are also available. These can range from little small pressurized CSTRs to rather exotic coaxial mixing systems with spinning bands and whatnot. These do have enhanced mixing, but they're operationally complex and they're rather expensive, and they're usually quite specific to the chemistry that you're going to use. But certainly have place, but where possible, static mixers are a simple and low-cost solution that apply for most chemistry, including most of the organometallic chemistries that you would be interested in for pharmaceutical processing. So this characterization of mixing that I mentioned is part of the overall theme of how we go about scaling in these platforms that we've developed that I mentioned on the first slide. So what you see here is a platform for plug flow. This is a very small laboratory scale that would operate in the order of tens of grams per hour, where we've got mixers that are characterized by our mixing characterization model, and also where we're characterizing other things like the feed pulsation. So that pulsation actually translates into stoichiometry as well, because if you have more than one pump operating and they're at different pulse rates, you can get slight differences in stoichiometry. So understanding all of the attributes that control process parameters and being able to characterize them across scales from tens of grams per hour up to on the order of hundreds of kilos per hour is important to allow us to have accurate scale down models that we can then translate into production. So tens of grams, hundreds of grams still in a benchtop hood, kilos per hour now in a walk-in hood, and then on the plant floor out to tens or hundreds of kilos. And this technology has been used by us so many times that we've built a kind of standard reactor system where we have a few parameters that we can change, like changing out the mixing method or changing out the feed method. We have a broad range of temperature control options here, and we can also change residence time and distribution as desired in this system. But it's become a platform that we don't have to rebuild and revisit each time, and it's now well modeled by the small scale ones. And this is actually a platform that we've not only built for ourselves, but have also provided to other clients and shipped for them to use in their facilities. And this is a pilot scale space filling model of it. So one of the questions that might come up and was certainly of interest to us when we began was how do we handle the high temperature and manage heat transfer in these very fast reactions? And here we've let this frost up so you can see the actual path of cold in this. Normally it's enclosed in a nitrogen blanketed bed. But what we have in the setup is the feeds are being cooled down to the desired reaction temperature. So in this case that you see in the picture, it's minus 30 degrees centigrade, and it's on our pilot system. So we're actually pushing through about 100 grams per minute and going from room temperature, 20 degrees C, down to minus 30. So dropping by 50 degrees centigrade by just crossing a small shell and tube heat exchanger. And here's an example of that shell and tube heat exchanger that can handle up to or beyond even 100 grams per minute. So those heat exchangers take the temperature down, and we're measuring temperature both at the input and the output. Take the temperature down, and then we hit this static mixer. And this static mixer is very tiny. So the entire transit time across the static mixer, which is shown here with this down arrow, is only 200 milliseconds. And the question is, do you bother to blanket that with temperature cooling capability? And we don't. In order to maintain isothermal reaction zone, you'd actually have to have a TRTJ of minus 150 degrees in this case. So a massive difference that wouldn't even be achievable. And it turns out that if you well characterize your process to understand the sensitivity of the chemistry and design this properly, you can do this without the need to even cool that. So we allow the adiabatic temperature rise to occur in the reaction and then control the product output from there. So this is the standard procedure for these organometallic reactions now is to pre-cool the streams that are entering using these highly efficient shell and tube heat exchangers where the process fluid moves through the individual tubes that are in this shell. And as you can see, you have access to huge surface area because of the nature of the small tubes. And so this is the way that we're able to control heat in these systems. So in the case of the Boc-amine lithiation chemistry, we wanted to maintain the reaction long enough of the lithium and the Boc-amine such that it would consume most, if not all, of the sec-butyllithium, but not so long that we would start to see significant decomposition of the lithium amide. So in this system, the key design feature was to change out one of the heat exchangers after the mixer of the sec-butyllithium and the Boc-amine, such that we could have some residence time, in this case 30 seconds, but not so long that it would lead to decomposition. The nice thing about these very fast reactions is you can do a lot of studies at the full scale without having to consume a lot of material. And that's exactly what we did down here. So what this chart is down in the corner is we're measuring temperature at very many points across this. So you see all of these round circles are temperature monitoring points. So we're monitoring temperature of the feeds, monitoring temperature of the output, monitoring temperature after the reaction, after the residence time, and then into the reaction with the electrophile where we're monitoring temperature again. And this one actually has the workup attached to it. And what I wanted to highlight here is in looking at this over the course of just a few minutes, we were able to explore 4 different conditions in terms of feed rates and really optimize on the fly the process and get to the point where the process is producing exactly the way we'd like it to. And we can do those studies on the actual system before we engage in extended run times. And that initial performance, when we were first tuning up the reaction at about 300 grams per hour and a chiller temperature of minus 50, gave a crude yield of about 80%. By tuning some of the parameters, we were able to lower impurities down from 10 to 15% for this major impurity down to 2%, and thereby increase yield up to 95% with high conversion in both cases. And the total flow rate through this system, the pilot system that I showed the picture of previously, is about 500 mils per minute. And we're able to monitor in real time all the mass flows into it. So this is the stoichiometry of the reaction being monitored by all the mass flows attached to the feeds in real time, as well as temperature at various points along it. We're also typically monitoring pressure at various points, and you can, if need be, add PAT at this at various points as well as a way to monitor product quality and reaction performance. So the development life cycle here is initial development on a 50 gram per hour system in a benchtop hood, and you see that here. From those lab results, we can inform any changes that we may make to the reactor design or configuration. And in this case, we designed the system to operate at about 500 grams per hour in the Kilo lab. And if it's an internal project, if it's a project that's going to be executed at Cambrex, we can use this equipment in our own facility or transfer it to any of the other facilities that we might be doing manufacturing at by truck. You build it, test it, and ship it. Or if it's being used by an external party, we can design the chemistry, build the equipment, and then ship that to their manufacturing site. So the other nice thing about this is we can do those studies at full scale for the final tuning of the process. So here you see the 500 gram per hour unit actually in a benchtop hood, and then transferred after factory acceptance testing, transferred into our GMP facility where it ran in a Kilo lab walk-in hood following IQ/OQ for GMP production. And the results of that was a 7-batch campaign where we were processing on the order of 4 to 6 hour runs, processing a total of about 650 liters of process stream to produce a product of about 23 kilos in that process stream with an assay yield, average assay yield of 86%, which was well in line with what we expected. And for each reaction, we have this extensive data, usually taking data points every 3 seconds on things like temperature, flow rates, and pressure throughout. Those of you that are paying attention will notice that there is a pressure rise at the end of this. This is why this process is run for on the order of 6 hours. With any organometallic, organolithium reaction, you're typically going to, at some point, form insoluble impurities due to the decomposition of organolithium species. However, as long as they're well understood and mechanisms are in place to determine pressure rises and control them, they can be managed even in these extended production runs. They can be managed in a number of ways. They can be managed with clean in place type technologies, or they can be managed with switching valves for the critical part of the reactor to allow 2 process trains. In this case, neither was necessary as we were running for a shift each time. And then at the end of the shift, we would clean out that system and get it ready for the next day. So this common reactor platform for organometallic transformation has been broadly applicable for us. We've used it in a broad range of chemistries, including a few that we've published on. And really, it's being able to take a really broad range of different types of chemistry onto the same platform, which builds up process understanding of the platform and really enables us to rapidly deploy it in the future. So here, alene gas was lithiated and then transmetallated to zinc, and then in the presence of a chiral ligand, asymmetrically added to an aldehyde, and that was published in 2017. More recently, we published this paper with the selective lithiation of dibromopyridine, and then the use of that lithium to add to an amide selectively forming a ketone. We've used not only gaseous reagents for the organometallic formation, but also quenching an aryl lithium with gaseous CO2 to make lithium carboxylates in systems where benzyne formation's a challenge. And then borylation chemistry is another very common chemistry that this platform's been used on. So with that, I'd like to move to the case study on photochemistry. And in particular, we'll talk about an asymmetric Atropisomer synthesis that was performed using a photochemical process. And here again, the same design principle where we have our iris photochemical reactor that can be applied for small scale batch screening. And that same light source is then used for our flow development at 100-watt scale. And then that translates to our 5 kilowatt production scale reactor. And this is a modular reactor that scales up in 1 kilowatt modules and can be scaled beyond 5 kilowatts. And what we've done here is we've held photon energy constant. So we're using identical chips across this. We've designed the boards and been able to apply those to all scales to produce the identical wavelength. We're able to use the same or enforce the same photon density across these because we control the design of the board and the spacing of the chips. And that means that we've got well-characterized photon stoichiometry, and we've characterized that as a function of power input as well. These are all variable power, so we can control the stoichiometry of photons at will. We've also been able to keep the same photon path length across scales, so that just means maintaining the same irradiated path depth. And so the tubing in these cases can be anything from 1/8 ID up to 1/4 ID tubing. And then characterizing the residence time and mixing across the system as well means that we can translate results from these batch screening at small scale up to production quite quickly. Just an example of that, as a trial, we developed a homogeneous version of this nickel catalyzed Photoredox cross coupling for arene emanation. Here we were able to replace what's typically an insoluble base with Dabco. We're operating at 457 nanometer light with a very narrow bandwidth for that light with a Rabipi catalyst at 0.06% loading and nickel at 5%. So on the small scale system, we could characterize things like the photon stoichiometry that's required and also the reaction temperature required, and look at the kinetics of the reaction as a function of the process parameters. And we're controlling here photo wavelength, photon stoichiometry, the reaction temperature, and time. We could develop the process such that at 50 degrees with 2.1 amps of input power for 10 minutes, we could get 73% conversion. For immediate scale up, we can run this fully automated and unattended in that small reactor over a 24-hour period to make, in this case, 82 grams of that isolated product that we could then use to supply downstream chemistry that we were interested in. And this automation and control of the system is enabled by our laboratory operating system that allows us to monitor reactions even remotely, and also include the ability to shut down safely reactions if something goes wrong. Because of the similarity in the reactor design from small scale to large scale, we could rapidly go to larger scale. And indeed, once this process had run successfully, we moved on to scale that up to the full scale photoreactor and ran that for 6 hours to achieve an 83% assay yield and made 1.1 kilo of this material. So this rapid scale up can take place over the course of days or weeks and not months, due to the alignment of process parameters with reactor parameters across this. So the approach to photochemical process development is somewhat different than it is to chemical development. You have all the parameters of chemical characterization that you need to do, where you're looking at things like solvent, catalyst, etc., reagent stoichiometry and whatnot. But you also need to study the photophysics of the process that you're running. And so understanding the molar absorptivity of the products, the reaction mixture, the starting materials, and then being able to use that data to predict the properties that the reactor needs to have, and then being able to design the reactor. And so bringing together reaction design and reactor design, we're able to come up with an optimized manufacturing process for photochemistry here. So the project that I'll discuss in some details, the development and design of this API intermediate via photochemical reaction for an atropisomer. So the initial process used an organic catalyst to effect easy isomerization of this olefin, which in the Z form would then cyclize down to form this carbon-nitrogen bond, which resulted in the formation of two atropisomers due to the barrier of rotation around this nitrogen aryl bond, which required chiral separation because this is a non-chiral process. It's a racemic intermediate that's obtained and then subjected to chiral separation by chromatography. What was noted was that the product, or the starting material one, actually had some photoabsorption, and there was some reason to believe that this easy isomerization could be affected photochemically as opposed to with a chemical reagent to go after this cyclized intermediate. So after characterizing the absorptivity of that molecule and determining that a wavelength of 460 nanometers would be appropriate to use in this easy isomerization, we could characterize the photostationary state of that isomerization as a 3:1 E to Z ratio. So the natural next step is to see, can we use some chiral catalyst to affect asymmetric cyclization of that Z intermediate down to the desired P atropisomer. And indeed, screening of chiral catalyst did reveal a catalyst that was able to catalyze this process down to give the desired atropisomer in 90% EE. There is an impurity that's formed along the way, and that's a dimer of these intermediates that is a 2+2 across the olefin that we'll have to deal with. And so studying the various parameters that are available to us, things like temperature, we can study the formation of the impurity as a function of temperature. We can study the impurity as a function of the power input. So in other words, the photon stoichiometry. And the system that we're using here is very simple. We've got a mixture of the catalyst and our olefin, amine, that is just pumped through the photoreactor in a single pass to give product. And what we observed is that the product purity increases with increasing temperature. And I'd note that this system, we have independent temperature control of the reactor, separate from cooling of the light source, which is necessary. And then product purity decreases with increasing light intensity. So here's why it's important to be able to vary the stoichiometry of the photons as well as other process parameters. And we can do that by changing the power on the system. The other thing that was observed is that increasing residence time led to decrease in the amount of product that was observed. And so the conclusions here were that the photoisomerization of the E isomer into the EZ photostationary state was a very fast reaction, that the major impurity was indeed a photochemical 2+2, and that excess light intensity resulted in increasing 2+2 dimerization, and increasing temperatures facilitated the second step, which turned out to be a thermal step mediated by the chiral catalyst that affected the desired cyclization. So this performance from the initial single pass operation did not really meet the purity or productivity requirements for the operation. And we decided that a recirculating feed would be a better process, because now we can separately affect the thermal cyclization process separate from the photoreactor. So now we are separating the two steps of the reaction, and we can optimize both somewhat independently by using a recirculation mode. This data that we have thus far suggested that the photoisomerization was actually rate limiting in being able to, the amount of Z isomer was actually limiting in these So now in a recirculation mode, we're able to decouple those two and achieve quite good conversion to the desired two, consuming the easy isomer thermally through this catalyzed cyclization. And also led to some reduction in the dimer product. We could further characterize that using DOEs. We can look at catalyst loading, temperature, and current. Current, again, being photon stoichiometry. This is a unimolecular reaction of one with a catalyst. So there's no other stoichiometry to vary but the photon stoichiometry. And monitor purity as a function of catalyst loading as well, and found that even at low catalyst loading, we had decent conversion, which was important because of an expensive catalyst, decent performance as well. And this was characterized not just for these parameters, but for others as well. And the learnings from the DOE allowed us to design the residence time for the photoreactor. And so this is how long is the coil that's being irradiated, the ratio of how big this holding tank would be to that photoreactor coil, what power we needed to use, and what process temperature we wanted across this entire system. And then from that, we could do on lab scale a 22-gram demonstration run. And here's the data output from that. It took a 10-hour reaction time. We achieved an 85% yield in the process. And we could characterize productivity both as grams per liter per hour or in terms of the grams per kilowatt, grams per hour per watt of energy input, so that we could understand the efficiency of this process. And since we have well characterized both this small laboratory system, both in terms of the energy output as a function, or the light energy output as a function of energy input, the photon stoichiometry, if you will, the spectral width of the wavelength that we're irradiating at, and actually the distribution of that light across this reactor array, where we have an average power, in this case at 50% of 14 watts per meter, 1400 watts per meter. And we can do the same characterization for the large scale reactor. And so now that we have these two pieces of information, we can then go and make a prediction about what the scale up would be. So that demonstration run on the small laboratory scale told us that at scaling up to the larger reactor, in this case, just using a single module of that 5 kilowatt reactor to process 360 grams, it told us that it would take us 19.7 hours. The model did. In actual fact, it took about 19 hours to process and gave similar performance as expected based on the small scale reactor. However, we were still plagued by higher levels of the dimer product than we want. And the productivity we felt could be improved. And the way we felt that could be improved is if we did a fed batch recirculation, where we're adding the substrate slowly over time, as opposed to having it all in there at the beginning. This is our chromophore, so that's the piece that's absorbing light. So by not having it all in there at the beginning, we can actually make better use of the light across the entire depth of the reactor, which is 1/8 of an inch in this case, because we're not absorbing all of it within the first 0.1 or 2 millimeters of that tube, because we don't have a high concentration of the chromophore in there. And that meant that we could have a slightly shorter reaction time by better light utilization. And we also predicted that we'd see a 5% decrease in the dimer impurity by doing this fed batch approach as opposed to all-in recirculation mode. And so what that looked like in our engineering run was up here in this picture. You see our reservoir. This is a 20-liter reactor, and this is actually being pumped over into the photoreactor. And in this case, we just had one panel operating, but in practice, for scale up, we were going to run 3 for this 2 kilo. So there's 3 photoreactor banks that is passing through in a parallel type fashion. So this is split and then passed through all 3 of them. and then recirculates back. And what we see in batch versus fed batch is an increase in yield of 10%, and as predicted, a decrease in the percent dimer. And we also saw an increase in productivity in this new process. So for this reactor, going from 55 grams per hour to almost 100 grams per hour. And you can see here that we fed over about 11 hours, and then after that, all of the material was in, and it was allowed to continue reacting until it had fully consumed all of the starting olefin. So the one thing that we need to think about is how we're going to run this. So one way to run it is to have 3 shifts and run it continuously, because it's a 13-hour reaction plus setup and breakdown. But we have another opportunity in our laboratories that are quite highly automated. And in this case, we wanted to be present during the feed, but at night when it's just consuming the rest of the reactant in recirculation mode, we didn't feel that there was any critical need to be present, especially since we have this lab operating system that allows us to monitor all of this equipment through a common server, and then actually communicate with that server and that equipment through a number of different interfaces, including remote interfaces if need be. The other thing I mentioned earlier is that we can institute all kinds of safety protocols to automatically shut down in the event of a change in the process. And that was indeed implemented such that we could be there for the feed, and then at night this would finish out the reaction and then stop itself. And the next day we would be able to come in and work up that batch. And that really facilitated a campaign mode where we were able to accomplish 3 batches of this a week. There's a first step that I haven't discussed here. There's the photochemical step, and then there's a white film evaporation to concentrate and crystallization and isolation of that material. So we were able to run this every day over 6 days, where we're running the photoreaction in 3 batches, 1 each day. Fits very nicely into this campaign mode, and in that way we were able to deliver out or process about 30 kilos of input material over a short period of time. And all of the data from this is captured for each run, and that data becomes very useful when we go back to mine that data for process understanding. And so what you see here is consistency in terms of the isolated yield and the batch size production on that Iris Max. This is the team that did that. And as I mentioned, we processed about 30 kilos of material in less than a couple months under GMPs for that intermediate. So what I take away from our conversation today is that it's this multidisciplinary team of chemists, analytical scientists, engineers, software and automation engineers, and mechanical engineers able to design equipment along with data-rich experimentation that really allows us to rapidly and with very low risk scale up complex chemistries like organometallic chemistry and photochemistry. So with that, I'll stop and open it up.
Okay, I'm going to jump back in right now, and thank you so much for that robust presentation. We're going to get to the Q&As, but first, just a reminder to everyone, if you haven't submitted a question yet and you'd like to, you can do that right now. And if we do not get to your question today within the hour, we will be responding to all questions post-event, so we will get to all of your questions. So let's get to them. Okay, I see our first question is, flow chemistry isn't just for pilot scale work. What is its value in commercial manufacturing?
Well, really being able to manage these really complex and exothermic type reactions at scale. So being able to take what may traditionally be chemistries that could not be scaled and scale them out to production. And sometimes that means that you're able to leverage lower cost reagents, shorter reaction routes, to your API, and also manage them very safely. So I would think that would be the biggest.
Okay, very good. We'll move on to our next question. Let's see, the question is, what other emerging technologies help accelerate drug development?
I would point really to the data-rich experimentation that we're doing. And one of the other things that we're able to do there is we're able to connect that data to algorithms that are optimization algorithms at this point. So being able to apply an optimization algorithm to help determine the design space for a process very rapidly in an autonomous way is a really neat technology that we're anxiously exploring.
Okay, excellent. Thank you. And the next question that came in is, how is Cambrex applying flow and other technologies to client projects?
As I mentioned up at the start, quite broadly to a range of different types of chemistries, everything from photochemistry, electrochemistry, to traditional type thermal chemistries, and also cryogenic chemistries. Although I would say, I would know we do take an agnostic approach. I mean, where batch works, batch is our first choice. But where needed, continuous manufacturing can be deployed as a tool for the process chemist to solve problems.
Okay, excellent. We have another question that came in from Andy. Andy wants to know, well, first he says, thank you for the wonderful talk. Can you talk more about the capability of your in-house operating system?
So we use it quite broadly from laboratory development all the way through production in our early phase GMP suites, where it's able to gather data from any piece of equipment that's connected to it every 3 seconds. It can be used, as I demonstrated, in automation so that we can run many reactions unattended, run processes unattended. But I think the biggest value out of it is the data that we collect from it, and then being able to connect that data to algorithms that are able to do further operations on that, either back into automation or for better process understanding.
Very good, thank you. I see another question that came in from Mike. Mike is asking, have you used, and I hope I pronounced this correctly, have you used any Raman or inline IR sensors? How are these configured? Did I ask that correctly?
Yes, both indeed have been applied, and at-line HPLC as well as PAT. And these are usually, if it's a batch reaction, that's a simple solution. If it's a flow reaction, then typically we're designing a flow cell or have flow cells designed that allow us to put inline IR or Raman, or in fact HPLC. And in the HPLC case, we're just using an off-the-shelf Mettler-Toledo easy sampler to get the sample, but then translating that to an HPLC automatically.
Okay, great. Next question is from Zachary. Have you ever used flow for microencapsulation to also control the particle size?
We have done some processes where we've made nanoparticles and also mesoscale particles using flow. And there, it's really about the mixing and the way that mixing is done. There's a number of different challenges there in terms of controlling particle size, but we've certainly done everything from a few microns across to up to 0.5 millimeter type. Those are quite esoteric projects. It's not typical, but we have done those, and I'd certainly be interested in talking to them about that.
Great, thank you. Thanks for the question. We have another one actually just came in from Deborah Ann. She'd like to know if there are any new exciting innovations to watch out for within the flow chemistry.
I think electrochemistry is where photochemistry was probably about 10 years ago. So you're starting to see more and more applications of electrochemistry. And electrochemistry, it's more complex than photochemistry, I'd say, because you have this third phase of the electrode. But it also benefits from continuous flow in that you keep the anode and cathode close together and avoid ohmic heating. So I think flow is enabling for electrochemistry, just like it is for photochemistry. But the chemical transformations of interest are much less well developed at the moment, and it's definitely a development challenge that I think we'll see evolve over the next period.
Okay, I think we have time for one more question, and that's going to be from Kenneth. The question is, for photochemistry, does exploring different solvents help?
Yeah, absolutely. I mean, some photochemical reactions have what is called a bathochromic shift, and you can actually change the wavelength of absorption by being in different solvents. Certainly things that are engaged in hydrogen bonding. Hydrogen bonding can be an important organizational component of photochemical reactions that are bimolecular. And so having a polar protic solvent in some cases is very important. It can also accelerate excited state proton transfer type reactions. So yeah, solvent is not an innocent bystander, particularly in photochemistry.
Thank you. And you know what? I'm going to sneak in one more question before we're finished with the Q&As. And that question is, does Cambrex have capability for validating flow process now? If so, where is the location for flow process validation?
Yes, absolutely. We've got a number of flow processes that run commercially, and those are fully validated process. Those would be at our plants. So Charles City, Iowa is one of our major plants, and there's a validated flow process there. Another one of our plants is in Karlskoga, Sweden, where we have a validated flow process there. And we're actually in the process of transferring additional flow processes to our Milan site as well. And also the facility down in High Point, North Carolina has extensive continuous manufacturing capability that they've used across the entire life cycle of clinical manufacturing.
Okay, very good. Well, that is our time for today. I want to thank you so much, Dr. Matthew Bio, for all of your time and information and sharing all of that with us. And I want to thank you, the audience, for watching. Till next time, we'll see you back here at Life Science Connect webinar series. Have a great day.
Great, thank you.